All right, Sanai, in the last class, we did about the enzymes, okay? See, we had done about the different types of enzymes. Okay, we had done what are the definitions of enzymes. We had done about the polysaccharide, rubisco, inulin, all this we had done. Yes?
Yeah.
Okay. So, we'll continue with the enzymes in this class. Okay. So, we'll learn something more about the enzymes. Okay.
Mhm.
All right. So let's start. Okay. So the last class we looked into the enzymes. All the features of the enzymes. Let us look into some properties of enzyme. Okay. Some of the properties of enzymes. So, first of all, in the last class, we had done about the activation energy. Activation energy is the energy that will be required to initiate or start a chemical reaction.
Yeah.
So, the enzymes, what they will do is that they will lower, okay, they will lower the activation energy. Okay, they lower the activation energy. Lower the activation energy, okay, so that they can efficiently carry out the enzyme functions. Okay,
Mhm.
then now another important thing, when we talk about the enzymes, these are organic organic catalyst.
Mhm.
Okay, they're organic catalyst. We have inorganic catalyst also. Organic catalyst. Enzymes are very specific. That means one enzyme will work for only one substrate. Okay, one enzyme works for only one substrate. This is very important. It's an important property. Specificity is an important property of enzyme. Now,
Mhm.
next, when the enzymes at high temperature, we will find denaturation of enzyme. Enzymes, that means enzymes are actually proteins. At high temperature, what happens? The proteins or the peptide chains will break, peptide bonds will break. Okay, and the enzyme will be destroyed, so that is called as denaturation of enzyme. So enzyme denaturation is observed at high temperature. Okay, now here whenever we are talking about any turnover, whenever a reaction uses enzyme, it gives a very high turnover. One second. High turnover, that means high productivity of product formation. Okay,
Mhm.
temperature is also specific to a certain pH. Okay, so pH is a also important factor, and enzyme activity depends upon the pH as well. Okay, these are some of the properties of enzyme. Now let us see how the enzyme functions occur. Okay, how the different enzyme functions occur. Now, if you want to see the enzyme function or the how the enzyme activity is carried out. Okay, how the enzyme activity is carried out. Like for example, let us see here. Very important. Okay, let us see here. Okay. So, when we talk about enzymes, okay, when we talk about enzymes, so here, what the enzyme do? The enzyme converts the substrate into product. Okay, substrate into product. So, what happens, any reaction, okay, any reaction to continue should have continuous addition of substrate, because once the enzyme converts the substrate, it is not, it is going to be like empty, it is not going to have any work. So enzyme remains, but the substrate gets over, and in this way the rate of the reaction comes down. So if we want to if we want to carry out the rate of reaction, that we should have a steady supply of substrates, so that enzymes are always busy converting the substrates. Is this clear?
Yeah.
Okay. All right. Okay, let's see here now. Okay, so let's see. So when we talk about pH, this is the, whether it is acidity as neutral, amount of hydrogen ions in a particular solution. Now, for example, an optimum pH, okay, an optimum pH is very important for the functioning of the enzyme, because beyond that or below that pH, the enzyme function will be affected. So a very specific pH is to be maintained. If the pH is below or above, then the enzyme activity, then the reaction rate will be severely affected. Now, enzyme pH has this particular uh, effect. When we come to temperature, temperature plays a very important role, because we know one fact that high temperature will carry out denaturation, denaturation of protein. Okay, it will carry out what? Denaturation of protein. High temperature. Every enzyme, you should remember, every enzyme works on an optimum temperature. If that optimum temperature is, suppose, for example, if the enzyme is working at very high temperature, then what happens? There will be denaturation. Okay, the enzymes will be destroyed. Why our internal body is 37 degrees centigrade? The reason, because now you see, inside our body we have many cells, and cells will carry out metabolic reactions. Metabolic reactions means many enzymes are required. So every enzyme in our body functions very appropriately when our body has 37 degrees centigrade. So I can say, in our body, this is the optimum, this is the optimum temperature. Okay, in our body, this is the optimum temperature, and it is this optimum temperature that helps the enzymes to function appropriately. Okay, is this clear?
Yeah.
Okay, next. Okay, now suppose, all right, suppose if the temperature is very high, 50 degrees centigrade. So what happens? At high temperature, there will be denaturation of enzymes. Enzymes will be broken down, because why? The peptide bonds, peptide chain within it is breaking down. Denaturation of enzymes. Then next, let's see here. Suppose, for example, if the temperature becomes very low towards freezing, then what happen? Enzymes will not undergo denaturation. At very low freezing temperature, enzymes are preserved. Enzymes are preserved. Very low temperature preserves the enzyme. Very high temperature destroys or denatures the enzyme. Is this point clear?
Yeah.
Okay. All right. Okay, so these important points that you need to understand its complexity. Okay, so how the temperature and the pH works in an enzyme. Okay, and one more important thing that you should always know, that enzymes are nothing but they are what? Enzymes are proteins. Is this clear?
Yeah.
Enzymes will be what? Enzymes are proteins. Okay. All right. Next, suppose, for example, let us say, suppose we have an enzyme here. Okay, this is an enzyme. Now, all right. Now, we usually have a substrate, and this is the active site. This is the active site. So here we have the substrate. In normal scenario, okay, the substrate will bind to the active site and will carry out appropriate enzyme functioning. But here, what happens? Sometimes in the medium, there are structures. Okay, there are structures which are similar to the substrate. They look similar to the substrate. They have structure also similar to the substrate. This is called as competitive inhibitor. Competitive inhibitor. Why competitive inhibitor? This inhibitor, if it joins to the enzyme,
Mm-hmm.
enzyme function will stop. Okay, so now this inhibitor and the substrate, they are of same structure, same shape, everything same. So both the inhibitor and the substrate, they compete for this site, which is called as active site. Both of them compete for the same active site. So such inhibition is called as competitive inhibition. Okay, competitive inhibition. Is this point clear?
Yeah.
Okay. Okay, let's see now. All right. Suppose now the enzyme will also have another side, another side. And one second. And we have, oops, sorry. We have here the we have here the substrate. This is the substrate. And we know substrate will bind for the active site. Okay, it can bind for the active site easily because there is no competitive inhibitor. But on the other hand, we see that the, okay, this structure you will see. All right, this is an inhibitor. This is an inhibitor, and it is not attaching to the active site. It is not competing for with the substrate to join at the active site. Rather, it is attaching itself at the base of the enzyme. And it is here that we call it as a regulator site.
Yeah.
Okay, regulator site. So in such scenario, what happens? This is called as non competitive inhibition. Okay, now what will be the difference in the functions of the two? Suppose, for example, competitive inhibition. When the competitive inhibitor binds to the active site, what happens? the rate of reaction will gradually slow down, and a time will come when the reaction will terminate. Okay, reaction will
Mhm.
terminate. But look at the non-competitive inhibitor. What happens here? The substrate will readily bind to the active site, and inhibitor is binding to the regulator site. Okay, non-competitive inhibition, allostatic inhibition. So what happens? The inhibitor is not competing with the substrate for active site. So it is telling the substrate, add how much you want, but I'll not allow the reaction to go on because I will not compete for the active site. Rather, it is going and attaching to the enzyme on the other side. Okay, even if it is attaching on the other side, enzyme activity will be affected. Enzyme will not be able to carry out its work efficiently, and the enzymes, okay, and what happened, all the enzymes and the substrate, okay, will gradually simply remain as it is. Okay, will not carry out any kind of reaction.
Yeah.
This point is clear, Sana? Any doubt?
No.
No? Clear?
Yeah.
Okay. All right. So competitive and non-competitive inhibition. Okay, competitive and non-competitive inhibition. Now, suppose, for example, Sana, let's see here. You are carrying out a reaction.
Mhm.
Suddenly, you see that the reaction, graph is coming to a standstill. Okay, it is coming to a standstill. That means it is coming, it is slowing down. Okay, you see the graph is slowing down. So, suppose, for example, okay, so competitive or non-competitive, the graph is what? Slowing down. In the system, you have the enzymes. Keep this in mind. In the system, you have the enzymes, but the graph is slowing down. Slowing down of the graph indicates that enzyme is not converting any more substrate. Did you understand the scenario?
Yeah.
Yes? Or should I speak once again?
Can you say it once again?
Yeah, okay. Fine. See, let's see here. You get a graph, okay, and in the graph it is written that the enzyme is carrying out its function efficiently, properly. A
Mm-hmm.
time will come when the graph will become flat like this.
Okay.
Okay, flat like this. And when we check on the solution, we come to know that there are many enzymes only in the solution. There are no substrates. So what will you do at that point?
Um, add a
Yes,
sub
what will you do?
add a substrate.
Yes. Okay, we will add the substrate. The
Mhm.
moment we add the substrate, all enzymes get activated and they start converting the substrate into product. So this point is clear, right, Sana?
Yeah.
Yeah. Okay. We have different type of enzymes. Okay, some of the enzymes names I will give here. We have oxidoreductase. Oxidoreductase. These enzymes carry out both oxidation and reduction. Oxidation and reduction. Clear?
Yeah.
Okay, oxidation and reduction. Next is, let's see, ligases. The main function of the ligase enzyme is they catalyze the joining. They catalyze, okay, the joining of certain molecule. Okay, ligases. They assist in the joining of certain molecules. Clear on this?
Yeah.
Okay. Next. Next is, let us see, nucleases. Nucleases. These are class of enzymes which will cleave or which will cut or destroy the nucleic acids. Okay, so we
Yeah.
have nucleases. Okay, so these are some of the important classes of fungi. Okay, classes of fungi. There are many different types, sorry, classes of enzymes. There are many different type of enzymes. Okay, but right now we just studied the different classes of enzymes. Clear on this?
Yeah.
Okay, all right. Let's see here. Okay. Um, all right. This point is clear, right? Sana, any
Yeah.
doubt here?
No.
Okay. We'll do one thing. How much have you done in the school this chapter?
We're mostly done.
Okay, 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 okay. All right. Okay. Okay. So come to small section. I just wanted to take that. Okay. Come to, okay. So have you understood enzymes?
Yeah.
Yes. In the last class, I dealt with the enzyme substrate complex, right?
Yeah.
Yes. Yeah. Okay. Okay. Now, what we will take down is a small introduction or a small note on the lipids. Sana, can you tell me what are lipids?
Um, they're like um another molecules,
Yes, the molecules, yes.
hydrogen.
The fatty acids, right?
Yeah. The-
They're
Um,
fats.
yeah.
Yes. They're fats, the fatty acids. Okay. So let us say lipids. Lipids are heterogeneous compound. Lipids are heterogeneous compound. Okay, these are organic molecule. Organic molecule. Okay, heterogeneous compound. And why they're called heterogeneous? They have different proportion of carbon, hydrogen, and oxygen. Okay, there will be carbon, there will be hydrogen, there is oxygen. Okay, different classes in combination, different combination. Some will have sulfur, some will have nitrogen. Okay, so lipids are, okay, let's see here. Lipids, when we talk about lipids, we need to know about fatty acids and glycerol. Okay, fatty acids and glycerol. Whenever we digest lipid, whenever we take lipid in our daily diet, lipids are digested by lipase enzymes. And these lipase enzymes, okay, what these lipase enzymes they do, they start the digestion of lipids. So after the digestion of lipids, the final compound that we will get is the fatty acids and glycerol.
Yeah.
Is this point clear?
Yeah.
Okay, understood, right? Fatty acids and glycerol, yes or no?
Yeah.
Yes. Okay, now there's a specific bond which is called as the ester bond, which will be formed between the lipid molecules. We'll have the ester bonds. Okay. ester bonds are formed between the lipid molecules. Okay, let us see. So we'll have glycerol and fatty acids. Okay, so classification of fatty acids. So now fatty acids. Fatty acids are made up of glycerol. One second. Lipids. Glycerol and fatty acids. Okay, glycerol and fatty acids. Clear?
Yeah.
Yeah. Okay, fine. So we'll see. Okay, so when we talk about fatty acids, okay, when we talk about fatty acids, so we'll see we'll have saturated fatty acids. We have saturated fatty acids and we have unsaturated fatty acids. Okay, two type of fatty acids. We have the saturated fatty acids and we have the unsaturated fatty acids. Clear?
Yeah.
Okay. Saturated fatty acids. When we say saturated fatty acids, they do not have double bonds. They do not have double bonds. Clear?
Okay.
Saturated fatty acids will not have double bonds. They do not have double bonds. So basically, they are solid at room temperature. Okay, they are solid at room temperature. Clear? They do not have a double bond, but they are solid at room temperature. Is this clear?
Yeah.
Yes. Okay, fine. Now, next, let us see here. All right. Now, when we talk about and these fatty acids, where they are generally found, they're generally found in animals. They're generally found in animals. Unsaturated fatty acids, they contain one or more double bonds. Okay, one or more double bonds. Clear?
Yeah.
Yeah. Uh, I'm sorry, Sana. There was somebody at the door actually.
It's okay.
Yeah. Okay. All right. Okay. So now see this saturated fatty acids. Okay. See, do not have double bonds. Okay. This thing you need to be very clear.
Mhm.
Okay. Do not have double bonds. Okay, they are
Okay.
solid at room temperature and they are found in where? Animals.
Okay.
Okay, all right, this is important. So we'll see. Okay, unsaturated fatty acids, one or more double bonds. Okay, they are liquid at room temperature. Okay, they are liquid at what? Room
Yeah.
temperature. So can you now tell me, Sana, what we are talking about? Give me one normal example that we eat on in our daily basis.
Um, of a fatty acid?
Yes, saturated fatty acids. See, is it ghee?
Yeah.
Yes, it's ghee.
Mhm.
It's dalda. Okay, and this is, see, when we say no oil, right?
Mhm.
Yes. Did you understand this?
Yeah.
Okay. All right. Now, fatty acids. So, see, Sana, this distinction that I have given you, no, this is very important. Okay. This distinction that I have given you, this is highly important. Okay. Because, I've given you point to point. Okay.
Yeah.
One or more double bonds. They are liquid at room temperature. Okay. Is this clear?
Yeah.
Then next, let us see here. Classification of lipids. Okay, classification of lipids. So we have simple lipids, compound lipids related. Those you don't have to go into much deeper into that. I'll just give you a few examples on that. Okay, just give you a few examples on that. So these examples, if you keep in mind, that is also more than enough. Okay, so let us see. Simple lipids. Okay, simple lipids. All right. So in simple lipids, like for example, we may have, like for example, let us see in simple lipids what we studied just now. Example, we will take animal fats, the fats that are found in animals. Okay, animal fats. Clear?
Yeah.
Okay. Then when we talk about oil, these will be plant oil. Can you give me an example, Sana?
Like sunflower.
Yes. Yes, sunflower. Yes. Yes, very good. Sunflower. Okay. Then next, we may also see, like for example, waxes. Okay, waxes. So examples of wax, very common. Beeswax. We use this in cosmetics. In medicine, it is used. In cosmetics, it is used. Okay.
Mhm.
Animal wax. Animal wax. Is this clear?
Yeah.
Okay. All right. Animal wax. Next. All right. Animal wax. Okay. Bee wax. Then ear wax. We have a ear wax, right?
Yeah.
These all are waxes. They come under lipids only. Clear?
Mhm.
Okay. All right. You've understood, right? Yes?
Yeah.
Yeah. Okay. Now we will take some examples of compound lipids. Compound lipids. Okay, let's see compound lipids. So, okay. Now, when we say compound lipids, Sana, what do you understand by the word compound lipids? What do you mean by compound? See, I told you simple. So, what do you understand by compound? Very easy. That's why I'm asking you.
Um, are they like
Yes. Tell me.
phospholipids and stuff?
Yes, phospholipids, lecithins, okay, lecithin, cephalins, sphingolipids, all these compound lipids. That means these, I can say them, these are complex lipids. Okay, complex lipids. Is this clear?
Yeah.
Okay. All right. So let's see here. Okay. Now, okay, fine. So lecithin. Okay, lecithin we have. Now, lecithin is found in alveoli. Lecithin is found in alveoli. And this lecithin, what it does is this lecithin forms a surfactant in the alveoli in our lungs. You know that, right? The alveoli are in our lungs.
Yeah.
Hmm. Okay. Okay. Understood, right?
Yeah.
Okay. So, you heard about the surfactant in our lungs?
Not the surfactant, no.
Yes, not you have not heard, right?
No.
Okay. All right. So surfactant means alveoli. Okay, these are alveoli. Around the alveoli, they'll secrete surfactant. Like this will reduce the friction. Okay.
Yeah.
All right. Next. Okay. So we have even cholesterol also. cholesterol also. Okay, then we have a class of compound which is called as the prostaglandins. These are always found in areas where there is inflammation. Clear?
Yeah.
Okay, so these are all about the lipids, about the enzyme activities. Okay, about the different type of enzyme activities. Okay, have you understood, Sana? Yes, any doubt?
Yeah, I understand.
Yes. Okay, tell me, have you done the structure of DNA and RNA in
Oh,
your
yeah. We're also
school?
learning it. Yeah, we're also learning it again in our HL class, like in more
Okay.
detail or something.
Okay, okay. You're learning it again, right?
Yeah.
Yeah, okay. So we shall we start that?
Yeah, sure.
Okay, okay, perfect, perfect. Okay. So till now, have you understood carbohydrates, everything?
Yeah.
Okay.
Hmm.
All right. So let's start. Okay. So all these are clear, right, Sana? Any doubt you have, tell me.
No, not really, no.
No. Okay. So shall we start with DNA, structure of DNA and RNA? Yes.
Yeah.
Okay. All right. Now, this you know that this is our cell. Okay. This is our cell. Inside the cell, we have a structure which is called as the nucleus.
Yeah.
And it was found inside nucleus there is a highly coiled structure. We call it as the chromatin fiber or chromatin network or simply chromatin. Okay, and then of course we have the nucleolus. Okay, now see this very nicely. Okay, now what happened? um Mr. What he did, what he observed, that there are some kind of acidic material. Okay, Mr. Okay, Mr. He observed that there is some kind of acidic material, acidic material. Okay, acidic material inside the nucleus, and he named it as nuclein. Okay, later this was renamed as nucleic acid. Is this clear?
Yeah.
Yeah. Later it was renamed as nucleic acids. And later it was found that, okay, later it was found that there is two type of nucleic acids. It was DNA and it was RNA. Okay, it was DNA and it was what? RNA. Did
Mhm.
you understand this?
Yeah.
Yes, it was DNA and RNA. So DNA, RNA are the two nucleic acids. DNA and RNA are the two nucleic acids. Clear?
Yeah.
Okay. So now, just try to understand this, Sana. Okay. Now, why they are called as nucleic and why they are called as acid?
Um, because it's in the
Yes,
nucleus.
yes, yes, correct. You're right. You're right. Nuclein because they are present in the nucleus, and acid because they were acidic in nature. Okay, they
Okay.
were acidic in nature.
Hmm.
Now, let us roll out our sequences. Okay. Now, this is what scientists had found out. This is a cell. This is the nucleus. Inside the nucleus, chromatin network. These chromatin network, they will condense during the cell division. Okay. We studied, right, cell division, Sana?
Yeah.
Yes. You remember cell division, mitosis?
Yeah.
Yes. So these chromosome will condense during cell division to form these chromatin network will condense during cell division to form a structure which is called as chromosome. Okay,
Okay.
chromosome. Then it was found that inside the chromosome, there is a highly coiled structure. And this highly coiled structure is nothing but they said it is what? DNA. So when they opened this, they found this kind of structure. I'll show you. Mm. Okay, when they opened the DNA, this is what they found. So now, have you studied about nucleosomes?
Um, uh, yeah, but I'm not very good.
Okay, all right. So this is what they found. Okay, they found that the DNA, this is our DNA, this
Mhm.
is DNA, and the DNA was wrapped around a protein which is called as histone protein. What is it called?
Histone protein.
Histone protein. Okay.
Yeah.
Hmm. Then when they further magnified, when they further magnified, they found that our DNA, okay, is, one second, our DNA is double stranded. Okay, our DNA is what? Double stranded, and in between the strands, we have the nitrogen base. See, so this is our DNA. This diagram, this depiction, did you understand, Sana?
Yeah.
Okay, our DNA in eukaryotes, DNA is always associated with a protein which is called as what? Histone protein. Clear?
Protein. Yeah.
Have you heard histone protein?
Yeah.
Okay, they're associated with what? Histone proteins always. So now, if our DNA, this is found in the eukaryotes, then in the prokaryotes, the DNA is associated with DNA is okay, it is associated with non histone proteins, which are chemically nothing but polyamines. Okay, is this point clear?
Yeah.
Yes. Okay. Then now, even before earlier, we knew the knew about the DNA. What is the structure of DNA? We didn't even knew that DNA was the genetic material. There were a lot of experiments that was conducted, and because of this experiments, what happened? They came to know that yes, DNA is the genetic material. Clear?
Yeah.
Okay. All right. Fine. Next. Now, now listen to this very nicely. Okay. Till 1956, even the structure of DNA was not known.
Mhm.
So they were basically two scientists. Okay, two scientists were there. And do you know the names?
Was it like mm,
Yes.
his last name starts with W?
Sorry.
I don't remember.
Okay, no problem. Um, one is a female. Her name is Rosalind. Franklin. Okay, Rosalind Franklin. She had taken a X-ray radiographic image.
Oh yeah, and she died of cancer, right? Because
Yes.
she was always doing X-rays.
Yes.
Oh, I remember.
She and yes, and there was another one that was Marie Curie, who
Oh,
was also exposed to X-ray. She's Rosalind Franklin. Even she died of cancer. Yes,
oh.
Rosalind Franklin. Okay, so she took an X-ray radiographic image. She
Mhm,
was not working with X-rays. Okay,
mhm,
all right. She was working with DNA. She took a X-ray radiographic image of DNA. She used X-ray to take the image of DNA.
mhm.
Okay, and following her was another person, Chorus Morris Wilkins. Even Morris Wilkins also gave the same structure. Now, their structure revolutionized the entire molecular biology. Why? Because later it was realized their picture was used by, okay, and Watson and Crick to take, okay, to deduce a very famous model which is called as the helix, double helix model of DNA. Okay, double helical model of DNA. Then later, Rosalind Franklin, Morris Wilkins, Watson, and Crick, all four of them received Nobel Prize for their immense discoveries in molecular biology. Is this clear?
Yeah.
Okay. Then next, let us see now. Okay, so what happened was now Watson and Crick gave the model of DNA. Okay, they gave the model of DNA and they gave certain postulates in the model of DNA. So, Sana, I'm going to teach you the postulates alongside. Together, I'm also going to take on the different features of DNA. Clear?
Mhm.
Okay. Now, according to Watson and Crick, okay, all right, okay. This is our DNA. According to Watson and Crick, first point that they gave us was that our DNA is a double helical double helical model. Is this clear? What you
Yeah.
understand by double helical model?
Like two strands that like
Helically
it's
coiled, yes or no? Yes, double. So it says that our DNA is what? Double stranded. So we call it as DNA. Now, SS. SS means single stranded. ssDNA is found in virus. Okay, our DNA is double helical. So we have two strands. But and most importantly, the two strands are anti-parallel, because if one is running from 5 prime to 3 prime, another one is running from 3 prime to 5 prime.
mhm.
Okay, they're anti-parallel with polarity. Polarity. So now we are going to study these 2 points when I actually do the placements of the sugar and all. Is this clear? Okay.
Yeah.
As of now, you know that they are anti-parallel and they are with polarity. Polarity meaning one strand is running from 5 prime to 3 prime, then another one will be 5 prime to 3 prime. Clear?
Yeah.
Okay. Then they said that DNA contain nitrogen bases. Okay, so they also said that the backbone, first of all, they said that. Let's see, constituents of DNA. In DNA, we have new, okay, before that, rather than that, I'll write DNA is called as polynucleotide chain. Polynucleotide. Poly meaning many, nucleotide, many nucleotide chains. Okay, now what is nucleotide? We have a sugar which is mostly deoxyribose sugar. We have we have phosphate. We have phosphate, and then we have the nitrogen bases. We have the nitrogen bases. These are the constituents of the nucleotide. Okay, nucleotide. So DNA will always have sugar, phosphate, and nitrogen base. And please remember, it is the phosphate that imparts acidic nature to the DNA. It is because of the phosphate group that the DNA is acidic in nature. Okay, then they said that the diameter of the DNA is 20 angstrom. Okay, 20 angstrom. Now, it will always have base pairs. We will always observe. We'll always observe what? Base pairs.
Yeah.
These are base pairs. Okay, so one complete helix, one complete turn. So I can say one turn, meaning one helix. One turn measures about 34 angstrom. Okay, 34 angstrom.
Okay.
And in one turn, in every turn, there are 10 base pairs, 10 base pairs, or simply BP, okay, per helix. 10 base pairs per helix. Now, this is the distance between one nucleotide and the other nucleotide. So distance between one nucleotide and other nucleotide is 3.4 angstrom. Okay, 3.4 angstrom. Is this clear, Sana?
Yeah.
Okay. All right. Now, so these were some of the features that were discussed on the DNA. Now, more are there. Okay. Now, let us see here. First, okay. So, first, what we will do, and yes, we will talk about the sugar. Okay. Now, let's see here. When we talk about the sugar, can you tell me, Sana, which sugar is present in the DNA?
Deoxyribose.
Yes, it is what? Deoxyribose sugar.
Yeah.
Yes, deoxyribose. What about in RNA? What sugar it is?
I don't remember.
Yes, ribose is very easy.
Oh yeah,
Rice
ribose.
sugar. Okay, so we are not doing RNA right now. We'll do DNA. So here the sugar is deoxyribose sugar. Okay, it is what? Deoxyribose sugar. So let's see here. Okay, so let's see here the sugar structure. Deoxyribose sugar. Okay. Okay, so this point is C. Okay, I'm not writing here all the time. C, H, OH, right? CH2OH. Now this here, one oxygen is attached to 1, 2, 3, 4, 3 carbon compound. This is called as furanose ring sugar. And this is deoxyribose sugar. Clear? The
Yeah.
sugar that is found in DNA. Okay. Ribose sugar, when we do the ribose sugar, or let me do it together. Okay. If DNA is double stranded, then we also have what? RNA.
Mm-hmm.
RNA is single stranded. Okay, RNA is single stranded. So
Yeah.
here we have ribose sugar. So let's see here. Sana, can you see any difference between the ribose sugar and deoxyribose sugar?
Yeah. The the C
Huh?
carbon.
Yes. Hmm. No, carbon is there in both. I have not written
The
because usually we don't write it.
H and OH.
Yes.
The H and OH.
Yes. Can you see this?
Yeah.
I can see this. This is carbon number two, a 2 prime carbon.
Mm-hmm.
Okay, 2 prime carbon. Here in deoxyribose, we have a hydrogen. Here we have an OH, hydroxyl group. Is this clear?
Yeah.
So, presence of this hydroxyl group makes the RNA more unstable. And this is DNA becomes very stable. Clear on this point? Yes?
Yeah.
Okay. Next is after ribose sugar, we have the pentose. Okay, we have the pentose. Yeah, instead of no, not pentose. Sorry, we have the phosphate. Okay, phosphate. The phosphate will give acidic character. The phosphate is always attached to the sugar. It is always attached to the sugar. Remember this. The phosphate will always be attached to whom? It will always be attached to the sugar. Clear or not?
Yeah.
Okay. All right. So phosphate and sugar together form the backbone of the DNA or the nucleic acids. Okay, backbone of the DNA or the nucleic acid. Now comes the nitrogen base. Nitrogen base are nothing but nitrogen compound. Their nitrogen base are nothing but what? Nitrogen compounds. Okay, or you can write they are compounds of nitrogen. So we have two compounds. We have purine and we have pyrimidine. Purine, these are larger compounds and they have two ring structure. Two ring structure. Pyrimidine, smaller compounds and they have a single ring structure. Let's see here. Okay, so purine will have one ring here, another one this. Okay, So two ring structure. Purine, this is pyrimidine. So purine are adenine. Okay, adenine, guanine, pyrimidine is thymine, cytosine, and uracil. Okay, so in the DNA, we will have, I'll write here G, we will have T, C, U. In DNA, we have adenine, guanine, thymine, and cytosine. Clear?
Yeah.
Whereas in RNA, we'll have adenine, guanine, uracil, cytosine. So what differs in DNA is thymine is present in DNA. Uracil is a characteristic feature of only RNA. This point is clear?
Yeah.
Okay. Understood or not? All right. So these are nitrogen bases. Okay. They are nitrogen bases. Clear?
Yeah.
Okay. Nitrogen bases. Then, of course, we did the sugar, we did the phosphate. All right. So together, they all make up the nucleotide. That is the phosphate, the sugar, and the nitrogen bases. All right.
Yeah.
Okay. Till here, any doubts, Sana?
No.
Clear? Sure?
Yeah.
Yes, you can have a look at the notes if you have written and just let me know. No, right?
Yeah.
Okay. So these are some of the compounds related to the DNA. Okay. Now, one more thing. Yes. The distance between the two nucleotides, the distance between one nucleotide here and two nucleotide. So I told you this is nothing but 3.4 angstrom. Okay, 3.4 angstrom. So here, when the DNA, helixes, we will find a major groove. We will find a major groove. And then a smaller groove will be called as minor groove. Okay, minor groove. Okay, now, all right. So DNA is double stranded and RNA is single stranded. Now, double stranded means pairing should be there. So, Watson and Crick, they included a rule which is called as Chargaff's rule. Okay, Chargaff, Erwin Chargaff gave a very important rule. Now, let's see here what is that important. Do you know what is that important rule? Yes.
Um, mm, I don't remember.
Yes. Have you studied base equivalence rule?
No, I don't think so.
Okay. All right. One second. Okay. Now, base equivalence. It says that in a DNA, the number of purine should always be equal to pyrimidine. Number of purine should always be equal to what? Pyrimidine. Clear?
Yeah.
Yes. So what do you understand by that?
Um.
Yes? Purine.
Yeah.
Okay. Before that, he always said that Purine first. Okay, we'll say purine always equals to bind with pyrimidine. That means if it is adenine, it will bind to, sorry, it will bind to thymine. Adenine will bind to thymine. Clear? And uh, let's see here. Okay, guanine will always bind to cytosine. I'll write it 1 second. Oops. Okay, see here. According to this, adenine binds to thymine and guanine binds to cytosine. This would be the perfect rule. Adenine will always bind with thymine by double hydrogen bond. Guanine will always bind with cytosine by a triple hydrogen bond. So Chargaff said that a purine can always bind with a pyrimidine, or a pyrimidine can bind with a purine. It can never be pyrimidine, pyrimidine, purine, purine.
Okay.
So if a purine binds with a purine or pyrimidine with a pyrimidine, that will be called as mutation. Okay, that
Okay.
will be called as mutation. So you understand what is mutation?
Yeah.
Yeah, what is mutation?
When a purine binds with a purine or a pyrimidine.
No, no. You can you explain me with what is mutation?
Oh, like it's when it's like
Yes.
there's like a change in the DNA
Uh,
sequence.
yes, it's a change. Yes, correct. It's a change in the DNA. It's correct. Yes, it's a
Um,
change in the DNA.
yeah. Um,
Yes, yes. It is a sudden change in the base pair of DNA.
mhm.
Okay, it's a sudden change in the base pair of DNA. Clear?
Yeah.
Yeah. Okay. So the sudden change is there. So let's see here the sudden change. Okay. All right. Now, see here. So it's a sudden change in the base pair of DNA. So that's called mutation. So according to Erwin Chargaff, adenine should always bind with thymine, and he said that guanine should always bind with cytosine. That means purine should always bind with what? Pyrimidine. Clear on this?
Yeah.
And adenine will bind to thymine with double hydrogen bond. Guanine will bind to cytosine with double hydrogen bond. Similarly, vice versa, thymine binds to adenine by double hydrogen bond. Cytosine binds with guanine by triple hydrogen bonds. Okay. Now,
Yeah.
this is okay. So this is Chargaff's rule. He also said that in a DNA molecule, the number of purine which is present, all total purine, okay, adenine, guanine, it is always equal to the number of pyrimidine. So that means in the DNA, how much purine is present, that much pyrimidine should always be present. Clear?
Yeah. Okay.
Okay. Yes. So these two are the deduction of Chargaff. Chargaff later gave the base equivalence. Oops. Base equivalence rule. Okay, base equivalence rule. Clear? Okay. According to base equivalence rule, adenine plus guanine and thymine plus cytosine is equal to 1. This is a constant value. This is what the base equivalence states. It states that adenine plus guanine, okay, divided by thymine plus cytosine is equal to 1. Or I can also say adenine plus guanine is equal to thymine plus cytosine. Clear, Sana? Any doubt?
No.
Okay. This is base equivalence rule. Yes?
Yeah.
Okay. Next. All right. Okay. So now when we talk about all these activities, okay, these all are deductions of the what do you call it, the Watson and Crick model. Okay. Now, one important thing that we all need to, understand here. Let me see what else nucleotides we have done. Okay, now just a small thing that we need to know here. Okay, now suppose, for example, we have a DNA molecule. Okay, we have a DNA molecule. Now, okay, all right. This is a DNA molecule. Let's see this very nicely. Okay, this is a DNA molecule. So
Yeah.
in the DNA molecule, we will have the sugar. Okay, we have what? Sugar. You see this?
Yeah.
Okay, we have the sugar. All right. Now, if in the sugar, this one becomes 3 prime, at this region becomes 5 prime. So, to this 5 prime, that means what I'm saying is, see to the structure. See to the structure. This is carbon number 1. This carbon number 2 I've given. Carbon number 3, 4, and 5.
Mm-hmm.
So phosphate will bind to carbon number 5. Okay, phosphate binds to carbon number 5. So we have a phosphate here which binds to carbon number 5. We have another phosphate that will bind to one here in the 3rd position, one here in the 5th position. Again here, one here 3rd, one here 5th position. Okay, so you can see that the sugar and the phosphate, okay, the phosphate is attached to the sugar either in the 5 carbon position or in the 3rd carbon. So now, Sana, you tell me here in this uppermost sugar,
Mm-hmm.
5 carbon is the one which is present at the top, or is it 3 carbon?
5.
5. Since either if phosphate will bond here or it will be vacant. In both cases, what happens? The 5 prime. In both cases, here phosphate binds with 5 prime. So this will be 5 prime. And here the phosphate bonds to the last one, that is 3 prime. So this is 3 prime. Now you understood why we have 5 prime and 3 prime?
Yeah.
Yes, this is clear, right? Why
Yeah.
we have 5 prime and 3 prime. Understood or not? Yes?
Yeah.
Okay. Now, the next section is in the opposite. So we have double stranded, right? So, 1 minute. Okay. All right. Let's see here. Now, here also, let's see here. Okay. One bond will extend from here. One bond will extend from here. So here we have a nitrogen base. Is this clear?
Yeah.
Okay. No, 1 second. Oops. Okay. So one ball will extend from here and one will extend from here. This is the next one. Okay. So one nitrogen base from here, one nitrogen base from here. So this can be either adenine by thymine or guanine by cytosine.
Mm-hmm.
See how the nitrogen bases are attached. See, like this. Can you see this?
Yeah.
Yes. Okay. So this type of arrangement will give in one turn 10 base pairs. Base pair means we are talking about the nitrogen base pairs. Okay, they are complementary also, base pairs. That means if this is adenine here, here will be guanine. Clear on this or not?
Yeah.
Okay. Now, listen to this very nicely. Okay. The bond by which the phosphate is attached to the sugar is called as phosphate bond. Okay, phosphate bond. The bond with the help of which the sugar is attached to the nitrogen base. This is N glycosidic bond. Glycosidic bond we know between the sugar, but N we are giving it's based on nitrogen. And the base pair, one base pair is attached to the other base pair, okay, by either double or triple hydrogen bond. So see, Sana, how many bonds do we have joining the entire DNA structure? Can you tell me?
Um, we have three.
Three. One is the phosphate bond, or we also call it as the phosphodiester bond. Clear on this?
Yeah.
Okay. All right. So this is how the DNA is, how the DNA are arranged. Okay.
Mm-hmm.
All right. This is clear. Any doubt here?
No.
No. Understood? No? This is how the
Yeah.
DNA will be arranged. Okay. Okay. All right. So this is the different type of bonds, all this that will be formed, that will be present in the DNA. Now, when we refer to the DNA, we refer as nitrogen base pairs. When we refer to the RNA, we refer as nitrogen bases, because RNA is what? Single stranded, and DNA is double stranded. Clear?
Yeah.
Okay. All right. So let's see here. Okay. Now, when we talk about when we are saying about the RNA, RNA is a ribonucleic acid. Okay, when we are talking about the RNA, ribonucleic acid, definitely this is what? Single stranded. Okay, single stranded. So we have different type of RNA. We have mRNA, we have tRNA, and then we have rRNA. These are the three types of RNA. RNA is usually the genetic material in viruses.
Mm-hmm.
Okay, we have different type of RNA: mRNA, tRNA, rRNA. mRNA is called as messenger RNA. tRNA is called as transfer RNA. This is messenger.
Okay.
This is transfer RNA. This is ribosomal RNA. Messenger RNA is the one that is actually taking part in the process of photosynthesis. Sorry, not photos, sorry. One second. Protein synthesis. Protein synthesis. tRNA also is involved in protein synthesis. tRNA, transfer RNA. This is the only RNA which is double stranded. It is coiled. It is okay. rRNA, ribosomal RNA. rRNA is mostly found in ribosomes. Okay, rRNA, ribosomes. A major part of the ribosome is also made up from rRNA. Okay, this is also made up from rRNA. So these are the three different type of RNA that we need to study when we do the protein synthesis, when we do the transcription. Okay, so all right. Okay, so this is understood. Yes.
Yeah.
Suppose in a pair we have adenine, thymine, adenine. So these are called as complementary base pairing. Complementary means if it is the thymine, it will be an adenine. All right. So complementary base pairing. Okay. So this is an important point that you need to note. Complementary what? Base pairing. Okay. So we have the RNA. RNA is not the genetic material. However, it is found in, like for example, in viruses. And the early earth, when the evolution of earth took place, that time the genetic material was RNA. Now RNA, if you enumerate the different features, the RNA will be highly reactive. Okay, highly reactive. They undergo mutation easily. Okay, they undergo mutation easily. All right. So if suppose for example, if we are storing genetic material, if any structure in our body is storing genetic material, that structure should be less reactive. It should be stable. This is unstable. And also that structure should resist mutation to the maximum. It should not happen that little bit of some changes are there, mutation can occur. No, it should resist mutation. See, even in our body, DNA replication takes place. Okay, DNA replication is taking place. There are many mutations that are being done, created. Many mutations are there, but why we are not affected? Because those mutations are removed before they actually come to the process of cell division. Okay, after the DNA replication, what happens? Proofreading is done. Whatever the wrong inserts are there, wrong bases, all these will be corrected in the proofreading. Only after that DNA will go for the process of cell division. Is this understood, Sana? Yes.
Yeah.
Yeah. Okay. So we have studied the structure of the DNA. We have studied the constituents of the DNA. Okay. We have studied what is the difference between the DNA and the RNA. We have okay. So double helical model we have done. Yes. So in
Yeah.
an upcoming class we are going to do on semi-conservative method of DNA. Okay, we are going to do a Michelson and Stahl experiment. All these experiments we are going to do. All right, and also we will talk about the process of DNA replication.
Okay.
Okay, all right.
Yeah.
So yeah, one more thing, Sana. I would just give me a moment. Just give me a moment.
Mm-hmm.
Yeah. Now, yes. Just give me a moment. One thing I'll mention here. After that, we can just sign off. Okay. So tRNA. This is also called as soluble RNA.
Mm-hmm.
Okay, and it is the largest one, and also it will have a structure. For example,
Yeah.
1 minute. It'll have structure somewhat like this. Okay. All right. This
Mm-hmm.
is the structure of tRNA. Clear? This is the structure of tRNA. mRNA will be linear. Ribosomal RNA, rRNA, this is also linear. Okay, all right.
Mm-hmm.
Okay, Sana, all right. Have you understood today's class?
Yeah, I have.
Okay, so shall we end the class here?
Okay.
Okay, then. All right, bye. Take care.
Okay.